Hello and welcome. This is probably the last time I will be saying that in this course because we are at the very end. Drum roll please. This is the last lecture of the course of Nanobiophotonics, where I will simply present a summary and I will revisit one or two topics, and thereby it will be adios, it will be goodbye from my side. So that after having complete knowledge, almost a substantial amount of knowledge, no knowledge is complete, you can go on in your own merry way and try to decide how to utilize this information that I have tried to impart you onto your future endeavors. So, what were the major goals of nanobiophotonics? The major goals of nanobiophotonics were actually understanding a disease at a molecular level, right. Thus far, the problem that medicine suffers from is that it has become too much symptomatic. You go to a medical professional, a doctor, because you have a fever or some other kind of ailment. The doctor measures the temperature, the doctor tries to do some test, and based on that symptom, clinical symptom, prescribes some medicine. But what exactly is your body going through when you have a fever? Now, almost all disease have some sort of a feverish reaction, right? Malaria has fever, dengue has fever, COVID has fever, sometimes cholera and diphtheria brings in fever, influenza has fever. So, can we not try to understand what exactly is happening at an individual molecular level to describe the disease or describe the fever is? Fever is common in almost all diseases, right? You get the similar amount of temperature, 101, 102 degree Fahrenheit and so on and so forth. But what exactly is happening at a molecular level in your body? Is it because of a pathogen has interacted? Is it because your own body is going through some kind of a immunal response? Is it something else? We need to understand the disease at a molecular level. Of course, if we are able to understand the disease at a molecular level, we have been able to detect the disease at a very, very earlier stage. At certain cells have had been detected, certain couple of cells have been detected, or certain subcellular organelles have been detected, and that will help us treat it much easily and in a much faster and rapid manner. And from that, can we prevent the disease from spreading? We localize the disease to certain cells only, and the worst case scenario, remove couple of these cells, your body is robust enough to replenish, to create new cells in its place, and then create it, heal it completely without having any scar left. And of course, a targeted treatment of disease. Suppose we have identified the presence of a particular pathogen in a certain corner of a cell of your body. We are precisely targeting that cell, that pathogen at that specific place, instead of sending medicine all over the body, either by injecting it through blood vein, through your veins, or by consuming tablets which will then get digested and then mixed with the bloodstream and whatnot, thereby spreading all over the body. Yes, we have got pharmacology has pharmacology has increased tremendously, and as a result we can precisely target certain areas of the body. But can we target individual cells inside a living dynamic body? So, these are some of the major goals that nanophotonics want to understand, want to achieve, and want to avail itself for that. And because in order to do that, we use a plethora of techniques. Bio-nanophotonics, nanobiophotonics borrows ideas and technologies from vastly different field. It is an interdisciplinary subject. So, we utilize spectroscopy tremendously, both absorption, emission and scattering. Scattering is Raman, emission is fluorescence, absorption is of course FTIR, infrared absorption spectroscopy. We use light microscopy and tomography. An entire module was dedicated for microscopy of the brain. You have fluorescence microscopy, near field microscopy, laser scanning confocal microscopy, optical coherence tomography, etc. Not only we want to see, image, understand, but we also want to interfere. We want to modify and we want to produce cures. And thereby we have light activated therapy, where you have photodynamic therapy, photothermal therapy, where you inject nanoparticles. These nanoparticles create some kind of a reactive oxygen species. Those reactive oxygen species either eat up tumor cell, or they can simply upon absorption of light, these nanoparticles emit heat, very localized heat, and this heat then burn the nearby area, thereby destroying pathogens or cells that has been affected by pathogens or tumors that has metastasized. Of course, you can directly put laser based tissue engineering, a high intensity good enough laser that could be utilized for contouring the tissue. You will see that in eye, LASIK surgery, similar type of other surgeries like hair removal, tattoo removal. We are utilizing it for IVF, fertilization, artificial fertilization, all of those things you can directly use laser. Then of course comes the very hot topic of sensors. We can have biosensors. I told you electromagnetically active nanostructures. They will have a specific electromagnetic response, and upon their interaction with a biological molecule, their response change, and the change of response is directly related to the species that it has detected. Thereby it can be utilized as a sensor. We have discussed a lot about DNA microarrays, biochips, and so called lab on chips. These microarrays are the future. We can have large number of areas with complementary DNA that will be attaching large number of external DNA. If it matches, then it attaches. If it does not match, if the covalent bond is not there, it can be washed away. So, you can thereby see from the fluorescence which one is quenched, which one is present. Those areas which are quenched, you can thereby say that a particular DNA species have been found, and thereby you can identify a particular pathogen. This has been utilized significantly. This has been utilized significantly in the last few years for large scale coronavirus infection detection. Then of course, you have optical manipulation and enabling techniques, optical tweezers. We discussed you can non-invasively pick up a particular molecule, group of molecule, a particular cell, group of cells, put it at any localized place. Nanotechnology and optogenetics, where you use light to modify genes of a living organism, right. Biophotonics, bionanophotonics find application in medicine, ophthalmology, LASIK, retinal angioplasty, OCT based detection, oncology, that is cancer detection, tissue based cancer diagnostic, tumor detection, analysis of biopsies. I told you we work directly on analysis of biopsies, tissues using Fourier transform infrared spectroscopy, dermatology, skin diagnosis, tattoo removal, hair removal, acne removal, rejuvenation of skin. You dry away the moisture content, thereby the skin become more robust, rigid, tight, and thereby you look younger. Fertility, laser zona drilling, IVF, in vitro fertilization, where you artificially fertilize the ovum and the sperm, and the zygote that is formed is then transplanted into the uterus of the mother, and thereby you have full reproduction, you have full pregnancy. Dentistry is also being used, dental diagnosis and dental laser surgery, where you can try to detect either oral lesions, some types of mouth cancer. You can also do dental surgery, and cosmetic surgery related to this is also possible. So, what is the future of biophotonics or future of nanobiphotonics? Optogenetics and neurophotonics is going to be my bet. So, in the future, overall neuroscience is going to take over. Neuroscience will be one of the prominent areas, because neuroscience is so diverse. All of you have heard of artificial neural network or computational neuroscience, trying to mimic the brain of trying to do calculations. So, neuroscience, if you have to ask me one area in general where more amount of investment, research and development we should be looking at, is neuroscience. And of that neuroscience, my bias, my opinion, these are not fact, these are opinions, opinion will be towards optogenetics and neurophotonics, where light is being used to understand brain, modified brain, mimic the brain. Of course, in vivo imaging, live imaging from molecular level to the whole body, whole organ, optical metasurface based biosensors. The biosensors should be more and more going to be sensitive. Single molecule detection, you just saw single molecule detection in the previous lecture, single molecule detection. Two-dimensional material are coming up very strongly, but the application of two-dimensional material in medicine is still a nascent field. It is still a growing field, and I have yet to be convinced that two-dimensional materials are something extraordinary when it comes to medicine and biology. Of course, they should be, but the research, robust research on this regard, application of two-dimensional material in medicine, especially optomedicine or biophotonic medicine, has to be done by large number of groups working very, very seriously, very, very cogently, and I am asking some of you to look into this matter. Of course, a combination of photothermal therapy, photodynamic therapy combined, it is purely therapeutic. Another thing that has recently come into my attention is differentiating stem cells. Stem cells, these protocells, well, technically protocell is not the right term, but pluripotent, multipotent stem cells, they can differentiate into different types of cells. Same stem cell can differentiate into liver cell, kidney cell, blood cell, skin cell, etc. Can you control it? Can you control it? The idea here is to utilize nanoparticles and try to control by various means the presence of nanoparticle either on the surface or inside those stem cells, and trying to control where it will differentiate into. So, that is something that is coming up, application of nanoparticles in stem cells and trying to see if we can control its differentiation. Of course, what we read in last module, nanobiotemplates for biodegradable implants. You utilize biopolymers based on nanobiotemplates and then use some sort of a biodegradable implant made up of biological material, bio-inspired materials, and you put it in the implant, some kind of a pacemaker or some kind of implant in head that has similar properties at the present day printed circuit board based pacemakers, but after certain time, I mean it is not that harmful, or it will be attached with the part of the body, and some after a later period it can simply degrade itself. So, what are the biophotonics research group? I have specifically put biophotonics research group because this give me more hit, more search than nanobiphotonics research group. I have divided it into both nanobiphotonics group abroad as well as nanobiphotonics group in India, right. So, if you are interested in nanobiophotonic research, I strongly suggest that you could look into the research being taken by research being done by few of these groups. By no mean I know them individually or I am recommending them. They don't need my recommendation. They are working tremendously, and some of their work are the very best. These are some of the groups that I follow to see what kind of research they are doing, and I can recommend them to you. If you are looking for higher education, if you are looking for PhD, or if you are just curious, some of the works done by these groups abroad are fantastic. There are equally good people both inside the country who are doing good work. I will let them. The next slide is for them. So, in abroad, J.C. Huang's group at NIST USA, National Institute of Standards and Technology, they work in bio-nanophotonics for molecular imaging. They are the people who are looking for single molecular imaging, single molecule detection, single molecular imaging. So, please follow this. Fujimoto group at MIT USA, Massachusetts Institute of Technology, Biomedical Optics Research Group and Biophotonics Group, Biophotonic Research Group at Ghent University, Belgium. I had a good fortune to visit Belgium and Ghent University, but I missed visiting their group. Leibniz University, Hanover in Germany, has this gentleman Hester Kamp for biophotonics. Howard group at Notre Dame, very good work. Try to look into their work. It's the variety that Professor Howard brings. It's not just bioimaging or PTT or PDT. It's a combination of several work. Nemoto group at Japan, Biophotonic Research Group. At the same time, Jana Neider's group at Institute of Nanotechnology, Portugal, where nanophotonics and bioimaging work is being done. They are working on diamonds, nitrogen vacancy in diamonds and whatnot. This is also very interesting. So, all of them is somebody I follow. You do not necessarily have to follow them. You can follow your own biophotonics research group. These are some of the one which is quite popular. They have large number of very interesting paper, interesting for me. So, this is just an opinion. This under no circumstances is an endorsement or advertisement of any particular group. I like this group because of the type of work that they have done. I myself personally find their work very interesting, and I am asking you to check it out. If you don't like, completely okay. You might find far better group which aligns better with your own interest. So, it is just a mere suggestion. Similarly, there is this group of fantastic research institutes as well as research groups inside India. I am a personal fan of Professor Maiti at Tata Institute of Fundamental Research. The work done is outstanding, no doubt about this. Professor Maiti's work at TIFR is outstanding. IIT Dharwad, IISR Kolkata has Professor Nirmal Ghosh and Professor S.P. Singh. Bio-optics and nanophotonics work is being done tremendously. Integrated Biophotonics Laboratory of S.P. Singh at IIT Dharwad is doing some very good work. Asima Pradhan at IIT Kanpur, her work is top notch. Then apart from certain groups handled by individual professors, you have a center, a laboratory, multiple professors, multiple researchers together. IIT Madras has this biophotonics lab at the Department of Applied Mechanics, and this is also quite interesting. MAHE, Manipal Academy of Higher Education, has a center for biophotonics. They have a center. So, multiple number of people, multiple groups working all together, complementing each other's work. And creating very good paper. One of their paper got published, I think, in Nature. I was very impressed by the work done at the biophotonics lab of Manipal. Then you of course have biophotonics laboratory at National Centre for Earth Science, Thiruvananthapuram, Kerala. I have less interaction with them, but I assume they are also at the very high standards. And finally, I would like to bring your attention to my own research group. I have my own research group of Nanobiophotonics at IIT Kharagpur, and these 4 or 5 people that you see, these are the people I have the privilege to call my student, and it's a completely interdisciplinary group of which I happen to be the PI. Shobhik is a PMRF fellow, also happens to be the teaching assistant of this particular course. Shobhik is from a physics background. I have, he is working on virus and pathogens, trying to utilize light for detection of virions, viral particles. I have Pranav, who is an electronics engineer, who is working on oral cancer. I have Karthik, who is a dentist, MDS doctor, who is also working on imaging. He is working on meningitis and pleural fluids and tuberculosis and whatnot. I have Shubhanita, she is a microbiologist. Her expertise are needed on a daily basis. She is also an expert in molecular dynamics. I also have Anagha. Anagha has recently moved, but her work is top notch. She is also from physics background, basically quantum mechanics background, and her work on both molecular dynamics, artificial intelligence, density function theory, as well as her expertise in biological matter is unprecedented. So, this is our group logo, this is our group website. If you want to know more about my own research, my own group, please visit this lab. You will have much more interaction, much more information. We are also quite fortunate to get funded by national institutes, national research bodies such as Science and Education Research Board, ACRB, Indian Council of Medical Research, ICMR, Department of Science and Technology, DST, so on and so forth. And this is where we exist. This happens to be my group. If you are interested in nanobiophotonic research, if you have your own idea, feel free to contact me. I would be available, and if you are so willing to pursue higher education, please feel free to go into the PhD entrance criteria of IIT Kharagpur. If you are eligible, I would strongly suggest you apply, and if you are able to clear the entrance exam, obviously like any other institute, IIT Kharagpur also has its own rigorous PhD entrance examination followed by personal interview, and from that, if you so choose, if both sides finds it's necessary, you can be among this group. This group contains students, colleagues, collaborators and partners whom I am privileged to work with. Every single person here is a gem, is someone interacting with someone, helps me increase my own knowledge. I get to learn from them rather than they learning from me. So, with this, I thank the course of Nanobiophotonics. I wish you all the best in your future. Thank you very much.